Okay. The question was, how about selective versus differential medium? All right. Selective, if I ask, what does it grow, what does it stop from growing, that's your selective aspect. Sometimes there's an overlap. If there's a pink versus yellow, and they both grow, say staph aureus versus staph epi, that's differential, tell the difference, both grew. Our blood auger, alpha, beta, gamma, they all grew, which would tell the difference, differential aspect. Okay? Little T tags, okay, probably be placed on different parts of the scope. Different parts. You would need to tell me what the parts were. Tell me what the parts were. Just go over it briefly. Pointers and fine. Okay, what are these? Those are your gesture lenses. But I want the power. If I put it on one, you got to say the 4x, the 10x, the 40x, or the 100x. You need to put 100x and more. Because that's the important one. This is simply your light source. In front here, you have something that turns back and forth. And as it shows here, it can open up the light going in on page 2.5 or close the light going in. That is, what is that lever called? That's the iris diaphragm lever. Iris diaphragm lever. Okay? That's what opens and closes. I know this is way back at the beginning, but it's still important. The outer knob is your coarse adjustment for big changes in magnification. The inner knob is the fine adjustment. This whole platform, everything sits on is called a stage. Lenses up here, you say, I want an ocular lens. Okay? Now, way in here is a teeny piece of glass buried in the stage. Piece of glass. That's called the substage, because what is it? It's like a submarine inside the stage, right? Substage condenser. It's called condenser because it doesn't really magnify, but it collects the light and condenses it so it goes up to your eye. Okay. Up here, the diaphragm is the iris diaphragm to let more or less light in. Fine coarse adjustment, light source, stage, what's this little teeny piece of glass inside? Substage condenser, can you just tell me objective lens? What are you telling me? Powers. Ocular lens. I mean, what else can you say? It's about parts of the scope. Yes?
Maybe, would you possibly ask what they do instead of
I could also ask what they do. Yes. You know, if there's knowledge there, I'm going to want to know if you know it. To know if you know it. All right? Take a deep breath. Come back to that. Okay. Gram stains. Okay, for simple stains, your positive and your negative stains. It might be easier to approach it from this pathway. You only had 2 negative stains. What were they?
Nitrocin.
Congruent. That's your only 2 negative stains. First, that means it stains the background and not the organism, right? Negative stain stains the background, not the organism. That means everything else is a positive stain. Everything else. Crystal violet, saffron, ground stain. That carbofuchsia used for the spore stain. Malachite green used for the spore stain. Steel stain organism. Positive stain the organism. So those are some of them. Natural and blue. Positive stains the organism. Negative stains the background. Now we're going to go for about 15 more minutes because I want to give you all time to do the practice questions and to look at things in the lab and ask these questions yourself. We did coagulase, we did staphylococcus, we said no to the gram stain. Here are the bacteria to know. Okay, E. coli. Very few are gram-negative, most are gram-positives. Capsule is a white halo on the outside of that cell. White halo. If you see a cell in a white zone, and I ask what the structure is, you say capsule. Spores, those are clear circles. Are they surrounding the cell or are they often inside the cell?
Inside.
Okay, can be outside, but if you're looking inside and there's something in there, it has to be spore. Please don't tell me there's a cocci inside the rod. Okay? That doesn't quite do. Hyphae. Don't forget your aspergillus. Don't forget your aspergillus. Hyphae are your threads for your mold. Okay? And aspergillus came up, gave you that big black ball, and came down on hyphae. Now, if you have a big ball coming down on hyphae, and it's not on oil, and it's green, or blue, or pink, or purple, and it's not on oil immersion, it's still got to be what? Doesn't matter, you color it any color you want. Your aspergillus is a black ball on a hyphae. You can color that any color you want. But it wouldn't be on oil immersion because it's too big, right? It would be a lower power. Oil immersion probably means, in most cases, Escherichia bacteria. If I'm showing you something on a high dry, chances are it's a eukaryote, right? Unless there'll be some exceptions to this when we do the protozoans. Okay, we did bacillus, we did microbiology, Listeria monocytogenes, you got Neisseria gonorrhea, the only gram-negative cocci you got. They form how? 2 by 2 by 2. They're different cocci. Okay? Staph aureus, here's your mannitol salt and your coagulase again. Catalase, staph versus strep, E. coli, etc. That mentions the good things. Recognize aspergillus. I'm going to show you some pictures in just a minute. Bacillus anthracis. Borrelia is a what? You have 2 of these. Tight twist. Borrelia and treponema is the other one. They're both those. Borrelia is a spirochete. Causes Lyme disease. So as you say Lyme disease, you think fatigue, rheumatoid arthritis, you think vector that bit you to give it to you is a tick.
Deer tick.
Okay? Tetani we talked about, mycobacterium we did. Know what the hyphae are under the scope. The mold hyphae are going to be big, right? You have it under oil, and they're tiny threads. You can barely tell. You don't even call them hyphae. It's a filamentous bacteria. We had one filamentous bacteria we talked about, and he's been back here, and I mentioned him. It was Streptomyces rhesus. Why did I talk about him? Because this is the antibiotic. Streptomyces. Okay? One filamentous bacterium. Streptomyces rhesus is on this list. It's right here, right here, right there. Okay? Right here. Okay? Right here. Oh, I didn't switch it. Y'all tell me if I switch it. I'm going to go back. Tell me. Tell me. Say, hey, that doesn't make sense. Okay. Brimstone. This makes sense, doesn't it? You won't embarrass me. I won't get mad. I will just make it better. That's all I'm going to do. Okay? If you do embarrass me, that's my problem. Got it? That's not your problem. That's my problem. Okay? You guys laugh. That's good. Capsule, spores, hyphae. Clostridium we did. Listeria monocytogenes. Neisseria gonorrhea, only gram-negative cocci, 2 by 2 by 2 by 2. Staph, here's your mannitol salt coagulase. Staph versus strep, here's your catalase. E. coli, says no slides. Aspergillus, I'm going to show some pictures in just a minute, but don't think he has to be black. He can be colored. He can be blue, he can be green, he can be pink. Bacillus anthracis, there's your Borrelia, Lyme disease. Forgive the fact that it was meant to be there. Spirochete. Tetani, that terminal spore. Fringens and botulinum, subterminal spores. What does that mean? Not in the center, not terminal at the end,
Near the
but
end.
in between. Cryobacterium we did. Hyphae for fungi, bacteria. You don't usually say hyphae for bacteria. You just say filaments. Okay. Listeria can grow in your ice cream. Okay. And actually live in your ice cream. Here's your volutins, big, long spiral. You probably would have to do what with him? Estimate? Length. Okay? Remember, streptococci or 2 cocci or a chain of cocci in a capsule are probably going to give you, if it comes from the lungs, pneumonia. Here's your Streptomyces graeseus. Alright? Treponema is your other spirochete. Probably instead of putting them in alphabetical order, I should put the spirochetes together. Yeast. Yeast, don't forget them. Big balls, not bacterial. Unicellular fungi. Unicellular fungi. They reproduce by what? Bud. And what is this instrument? Back over here is the spec 20. You could have graphs off of that. Remember? Your optimal density versus cell number, percent transmission of light. Percent transmission, you don't have to put a light, but some people can think. Versus cell number. One graph's going to go up, one's going to go down. Know the difference? The measurement is based on what? Dry weight?
Turbidity.
No, it's based on
Turbidity.
Then two types of turbidity are optimal density and percent transmission. It won't be right if you tell me the measurement is based on optimal density. You gotta say turbidity. Turbidity. I want to go to some pictures. Really want to see some pictures. A lot of it's like, would it bother y'all if I turn the light off over again? Would that interfere with your lights? I'm going to stop and go a few times to do these test questions and look at slides if you want to. Syphilis. Okay, treponema is syphilis. Question, what is treponema called? Syphilis. Yeah. Okay. Somebody has a sense of humor. Somebody has a sense of humor. This is Greek alpha, Greek beta, Greek gamma. So, green, dirty green. See what's dirty green here? Dirty green here. That is alpha. I won't give you a plate and say, look on the plate. Fine. I'll have an area circled. And inside the circle is what I want you to answer about. Beta, if you can pick this up and look at it, you can see straight through to the other side and even read words on the other side. Gamma, gamma just means what? Blood all the way up to the growth, nothing happened. Nothing happened. Okay, the next one. This side, could that be staph epi? Yes. What would this be? Staph aureus. Staph aureus. Chocolate agar. You put the oxidase test, you're hunting for a non-pathogenic Neisseria. See that black colony? Black colony? Black colonies here. Those are oxidase what? Positive. They are probably therefore non-pathogenic Neisseria. The white ones or gray ones are what? Everything else. Okay? Staphylococci, just to help remind you of grouping. It always says grape-like clusters, but just think clumps of staph everywhere. Clumps of staph. Okay? Clostridium. This is Clostridium perfringens. Just close this. What you can do is you go to Google, put in, I'm going to Clostridium tetani found. Go down to Clostridium tetani, hit search, don't go to the web, go to images. Okay. This is Clostridium tetani. Looks like the drumstick, all the way to the end. There's no space here. See what I'm saying? It's on the end. Fringes would be a little bit back. A little bit back. Alright? Okay, I also want to show you some Corynebacterium. Same thing. Actually, we have one in here, that's all right. Anything goes. Where's my chaotics? Chaotic. Anything goes. Anything goes. Now, I want to give y'all time to look at your notes and to ask me to go over something you would like me to go over versus just sort of talking to you. Is there some subject y'all would like for me to go over? Would you like to explain a little bit better? Explain a little bit better. Not sure? Don't be shy. Well, I'm shy, but that's a weird thing for a teacher to do. Okay? All right. 2 questions here. I would do. I'm not saying they're on the test. I'm not saying they're not on the test. But they give you some more practice. Slides as far as you can see. Plates you can see. Equipment and spec twinning you can see. Alright? There's a growth curve up here. You want to give an example of growth curve, but we want to be on semi-log paper when y'all see them. The curve is going to be a real log paper. Alright? If you want to practice counting, you can pick up a plate and practice counting. And anything else I can think of. There's a waggling chest up here to look at if you want to see that. And anything that helps. Any questions? Okay, I would definitely do these 2. Let's try it for you. And I'll answer individually. If you don't want to answer in front of everybody, I'll answer one of them. Yeah, okay, private. Go ahead and you have time to look.
So you'll post that?